Okay, welcome back to my YouTube channel. This is the Webster Chileshe, your tutor in mathematics and sciences. I've come back with you again with the revisions in biology 2016. Okay, so before we go into the details, don't forget to like, share, as well as subscribe so that you become always the first person to be notified that I've posted a new video. Okay, we get into details now. Watch the video until the end. We're doing the whole part of section A, so I really want you to watch until the end of the video. You hear what I have for you. Okay, so question 1. Figure 1.1 below shows figure 1.1 below shows an animal cell seen under electron microscope. Now, if you can see this type of a cell from the diagram, you are able to tell that this is an animal cell because it doesn't have the cell wall. Okay. Identify the labeled parts F and J. So we have part F and the part J. So F and J. Where is J? Not really J, but G. Okay, so identify the part labeled F and G. So the part labeled F, which is this part here, that is the rough endoplasmic reticulum specifically. And then G, that is the nucleus. Okay, that's a nucleus. Okay, specifically this is pointing the nuclear. Okay, it is pointing outside, yes. Okay, very good. So this is the nuclear membrane. It is not really the nucleus, that is the nuclear membrane. So G is the nuclear membrane. But if it has pointed inside, you could have said that's the nucleus. So because it is pointing outside, that is the nuclear membrane. Then if you look at part H, that is the mitochondria. Part I, that's a cell membrane. Okay, so we go to question B. Explain the functions of the parts labeled H and I. So part H, we say that's the mitochondria. Mitochondria act as a powerhouse because that is where the cell, cell respiration takes place. So the function of the part H is to enable the process of cell respiration to take place. So respiration takes place in the mitochondria to produce energy. Then I is the cell membrane, which act as a selective permeable membrane, which select the substances that goes out and those which comes into the cell. So it acts as a selective permeable membrane to substances that enters and leaves the cell. In animal cell, it also provides the shape of cells and protects also the cells. Okay, but specifically here we're talking about it playing a role of selective permeable membrane, which select the substances to go in and out. Okay, so that's what you write here. Then you see, suggest 2 cell parts which would be present in figure 1.1 if it was a plant cell. So if this was a plant cell, you expect to have the cell wall outside. You also expect to have some chloroplasts. You expect a large permanent vacuole. Those are the permanent features you can expect to see in the plant cell. So you can pick from those. We just wanted 2 here: the cell wall, the chloroplast, and others. Okay, we're done with question 1. We now go to question 2 from enzymes. Figure 2.1 shows the effect of pH on the rate of enzyme catalyzed reaction. So we have reaction K. If we can see this reaction K, we also have reaction M. Okay. Now, what term is given to pH at J and L? The pH at J and L, this is the optimum pH. So that's the optimum pH, which is optimum pH at which the enzymes works well or works best. So that's the optimum pH. That's the answer for question here, question A. So that's the optimum pH. Explain why the rate of reaction K and M goes down after point J. and L respectively. So the rate of reaction goes down after the point J and L, because after that point the enzyme is denatured. When the enzymes, when the pH surpasses the optimum pH, then the enzyme will be denatured or destroyed. So it is because the enzyme got destroyed when it reached the optimum pH at J and L. Okay. Then we go to question C. Question C. Name the regions of the alimentary canal where reactions K and M take place. Now, the alimentary canal is the tube which runs from the mouth to the anus. Now, we want to identify where these reactions are taking place. We have two reactions here. We have reaction K and reaction M. So if you study carefully, where it is written in pH concentration, this is a pH scale, which is representing the acidity or alkalinity of the substance. So starting from 7 here, going this side, these are acidic, meaning that this reaction is taking place in an acidic medium. Then starting from 7, going this side, this is alkaline, which means that this reaction is taking place in alkaline solution. So, and moreover, if you look at this reaction, which is on the first part here, reaction K, it is taking place well in acidic medium, and is specifically with the highest pH of about 2.5. So, the reaction or the organ, the part of the alimentary canal where enzymes work well in acidic medium is the stomach. So, reaction K takes place in the stomach. Reaction L takes place in the mouth, or you can pick the small intestines between the two. Okay. We go to the next part. Suggest one enzyme which can catalyze reaction M. We go back to the diagram. Reaction M, we're saying that it takes place in alkaline medium like the mouth and the small intestine. So if you had picked the mouth, then we're going to use salivary amylase. But if you had picked the small intestines, you can use pancreatic amylase and many more. Okay, so we can pick more from that. Then apart from that, reaction M takes place in, okay, sorry, the battery is low. Okay, thank you. That's connected. So reaction M occurs in the small intestines or the mouth. So if you have picked in the small intestines, you can use the pancreatic amylase and many other enzymes which you can remember there. Then if you have used the mouth, use the salivary amylase. Okay, then I didn't say write both of them. I'll just give you an example. Here they say name one enzyme which can catalyze reaction M. So reaction M, talk about pancreatic amylase. If you have picked the small intestines. If you had picked in question C, you have picked M for mouth, use salivary amylase. D, Roman number 2, state one food nutrient which can be catalyzed by reaction K. So we said this reaction K occurs in the stomach, and the food which is mostly catalyzed or digested in the stomach are proteins. So the answer here is proteins. Okay. Next, state two factors that other than pH which affect enzyme activity. So we have temperature. Okay, excuse me. Temperature affects the enzyme activity. Apart from temperature, you have the enzyme inhibitors. Apart from the enzyme inhibitors, you talk about the enzyme concentration, talk about the substrate concentration. You just pick two out of those. You just wanted two. So you write them here. Okay, the enzyme inhibitors, the temperature, the pH, the concentration of substrate, the concentration of enzyme, many more. We are done with question 2. Go to question 3. Figure 3.1 shows a cross-section through a stem of a plant which has been previously dipped in a red dye for 6 hours. Okay. So if you can see this diagram here, yeah, under transport system in plants. Identify the parts labeled N, P, and the Q. So part N, that's a cambium. Then part P, that's a phloem. Then part Q, that's a xylem. R is the cortex. Suppose they ask that. R is the cortex. Then O is the pith. Okay. Now, which labeled part in figure 3.1 would be stained red? So which labeled part would be stained red? Remember, this was dipped into a red dye. So the part which will be stained red is the xylem, which is part Q. So the answer here is part Q. Give reason for your answer. Because xylem conducts water or materials upwards the plant. So when this plant was dipped into, okay, I'll do the demonstration here. If this is the plant we're talking about, which was dipped into the red dye, okay, those are the roots going upward like that. So we imagine that's a xylem. Then apart from the xylem, we also have the phloem. Okay, so we also have the phloem coming downward like that. The xylem is taking materials going upward, the phloem is taking materials downward. You see that, eh? So when the plant is dipped into the red dye, then you will expect that red dye to be absorbed from the roots and taken upwards the plant through the xylem cell, because the xylem is taking materials up the plant. So part Q will be stained here. Okay. That's the reason. This is because xylem is the one which conduct materials from the roots upward the plants. Conduct water, mineral salt, and many others. Okay, now we go to B1. Suggest 3 factors that would increase the rate of movement of the dye in the stem when it is dipped in the red dye. So here in short, they are asking you to list down the factors which affect the rate of transpiration, of which we have temperature. We can talk about temperature. Apart from temperature, we talk about low humidity. When the humidity is low, you expect the rate of reaction to increase. Even here, temperature is high temperature. High temperature can increase the movement of the red dye. Then apart from that, since they wanted 3, can talk about windy, light intensity, high light intensity, all those. Just pick 3, the factors which might affect the rate of transpiration, because transpiration is the process by which these red dye will be transported from the roots to upwards the plants. Name the process that will cause the movement of the red dye in the stem. Transpiration. Okay, I already spoke about it. Transpiration, suppose you are not aware of it, is a movement of the loss of water vapor from the atmosphere into the atmosphere, through the stomata. That's what is called transpiration. Okay. Question 4. Figure 4.1 shows a certain type of flower. Okay, can you see that? Now you can play around with the flower. Before even read the questions, you must be able to identify the parts. So we have part S, which is the anther, part T, which is the stigma, U, that's the ovary. Identify the parts labeled STU. So S, I said already, is the anther, T is the stigma, U is the ovary. Okay. Now B1, identify the types of pollination that occurs in the flower in figure 4.1. What type of pollination takes place in figure 4.1? So the type of pollination which occurs here is windy pollination. There are basically two types of pollination, windy pollination and animal pollination. So we have windy pollination and the animal pollination. So in this case, this is windy pollination. Give two features from figure 4.1 which supports your answer. So why did I say that here it's windy pollination? That's what they're asking you. Why did I say that this is windy pollination or windy pollinated flower? So, and the reasons are simple. It's because I have seen these, anthers, large anthers, and they're dangling outside, or they're hanging outside the flower. So in windy pollinated flowers, they have large anthers which hangs outside the flower. Okay. Then apart from that, since we say two reasons, apart from that, I also have seen this part, which is a feathery stigma. A stigma which looks like a feather, you see. So in windy pollinated flowers, they have large feathery stigma. So the stigma which look like feathers, and they hang outside the flowers. Okay, those are the two reasons you can give here. Apart from that, they are small. Flowers which are wind pollinated flowers, usually they are small. Okay, now apart from that, we go to question C. State one other type of pollination that other than the one mentioned in B1 above. So apart from the wind pollinated or the wind pollination, what other type of pollination is there? The other type of pollination is, as I mentioned earlier on, is animal pollination. Okay, you can finish that. It's animal pollination. So animal pollination is the other type of pollination other than the one mentioned. State two characteristics. Okay. State two characteristics of the flower where the type of pollination mentioned in C1 above occurs. So the type of flower where animal pollination takes place, what are their characteristics? The characteristics of animal pollinated flower, one, they have, large flowers. Large flowers. Flowers are big, usually, okay, in most plants. Flowers are big. Now, apart from that, the unique which makes it suitable for animal pollination are, one, they have a scented flower. They have a unique scent which makes insects to be attracted to that. Apart from that, they have colored petals. The petals are bright, which attract insect. Okay, not only insect, even human beings. Human beings who are attracted by flowers, especially women. Okay. Then apart from that, they also have nectaries, which attract insect like the bees and the butterflies. They search for those nectaries, for their benefit. So those are the characteristics of animal pollinated flowers. Apart from that, the anthers and the stigma are found inside the flower. They are not really found outside like these which we saw here. They are found inside the flower. Okay. Apart from that, they produce very sticky pollen grains. They produce very sticky pollen grains. Okay. They have small anthers, small stigma. There are a lot of characteristics of animal pollinated flowers. You just have to pick from the one I've just mentioned and put them here. Okay. Okay, question 5 coming from genetics. Question 5, genetics. Figure 5.1 below shows the paired degree diagram for two families A and B, outlining inheritance of a sex-linked disease called hemophilia. It's very important on this question to study the key closely. Okay, now let us study this key together. If you see this circle which is not shaded, it's a normal female. If you see this rectangle box here which is not shaded, it's a normal male. Then the circle shaded, that's a hemophilic female, and the box which is shaded, that is the hemophilic male. Okay, so from the study, now let us go to the question. Let's go to the question. family A and family B. Can we see that? So these two are the parents. Okay. Okay, I want to change the color. Okay, thank you. So we have family A and family B. You can see that, eh? Now, if you can see from these two, the mother and the father, they had 4 children, and one of them was hemophilic, which is a male. Here again, the mother, the father, they had children, and none of them is hemophilic. Okay. Now the questions follows. Question A, 1. From 5.1, which family has a parent who is a carrier for hemophilia? Family A has a parent who is a carrier. Family A has a parent who is a carrier. Now, why have we said this? It's because when these two crossed, one of their children had what? Hemophilia, meaning that one of these parents was a carrier. Okay. Okay, that's wonderful. Now we go to B, A Roman numeral 2. Give a reason for your answer. Oh, I've already given the reason, isn't it? It's because when these two parents were crossed, they had the child who is hemophilic, which represents that one of them is a carrier. This is hemophilic. You can see that. Okay. And then we're going to go to B, using the symbols, that symbol given, which is the capital H and the H on top, and the other one. State the genotypes of for offspring H and J. If J is a carrier for hemophilic or for hemophilia. Okay, so the symbol for H, since this is, remember that box shaded is a male who is hemophilic, so that will be, you write X as well as Y. Okay, so you're supposed to pick up theta Y. Now, because this H is hemophilic, this H must have a small letter H for hemophilic. So this is a genotype for offspring H. Now, what about J if she is a carrier? If J is a carrier. Now remember, on the key we have been told that a circle is male and is female, and if not shaded, mean that it's a normal. So in this case we are going to have these genes. Okay, let me rub this. Now we go for J. If J is a carrier, so remember that's a circle, so it's male. In female, in the last video I said for females it is XX. Now, because that female is a carrier, we're going to have the first X with a capital letter R and the second X with a small letter r. This represents a carrier. This offspring is normal, but he is a carrier of hemophilic. Okay. Okay. Now we go to the other part. Using a genetic diagram, show whether the offspring would be hemophilic or normal when H and J are crossed. When H and J are crossed, so would the offspring be normal? Okay, do I have a space down here? No, don't have. Okay, just right there. They have a very small space here. Okay, so we draw the cross diagram, a genetic diagram. So remember, we said the H has this genotype. Okay, let me write small things. You don't have space. Okay, so that is J is hemophilic. We have a small letter R. Then for the other J, the other one, this is for H, then this is for J now. You need to write small things, don't have space. Okay, so we have the small letter x, then capital letter R there and small letter R. Now because these two are going to cross, so we cross this one and this one, we have the x. Always get with the one with the capital letter first, so that becomes capital letter R, x, small letter R. The same letter again with the other one, we're going to have x, of which we are going to have now small letter R, small letter R. Next we have this one and this one, we have x. Okay, we have x, so x with a capital letter R, and y does not carry anything, remember that. Okay, it's very important to know that. And then the same again with the x, of which we are going to have okay, I rubbed that by mistake, so we're supposed to have that. Let me just start all over because I'm rubbing these things. Okay, so okay, I hope you're getting the concept here. So I will draw from here. I'll start from here. So we have x for h, small letter R there, and the y. Then we also have x for a female who is a carrier, so we have capital letter R and a small letter R. Then you start crossing, multiply this. X always comes first with a capital letter R, then x with a small letter R. So write the small letter, capital letter R there, small letter R there. The same x again with the other one. We are going to have x both with the small letters R on top. And then next we have this one here and this one, of which we'll have okay, so we have x, small letter R and y. Okay, can you see that? Now next we have okay, next we have the same. Okay, sorry for that, so I'm trying to draw there. Okay, and then we have the letters x, y. Now this x will have a small letter R, the y won't have anything. So that is the genetic diagram which they wanted you to draw. You always have to show, as I've been saying in the other videos, the F1 generation genotype, the F as well as apart from the genotype, also the phenotype. So this is the F1 generation genotype, the genetic constitution. But then for the phenotype, we will have one normal. Okay, yes, we'll have one normal. No, I left out something here, isn't it? When I multiplied this one times this one to be a normal. Okay, so this part isn't supposed to be like this. This was capital letter R. When I multiplied the y times the x, the first x here. Okay, so that's all for today. I hope you have enjoyed the lesson. Please continue going through biology revisions questions and watch them thoroughly, especially before the exam. It will pay you. Okay, please don't forget to subscribe, like the video, and you can leave some comments in the comment section below, and then I'll be happy to respond to your queries. Okay, thank you. Have a good day.